I am Dr. Silvan, Assistant Professor, PG Research Department of Biochemistry, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I am going to present the topic mitochondria. Mitochondria are membrane-bound organelles present in the cytoplasm of all eukaryotic cells that produce ATP, the main energy molecules used by the cell. Mito means thread, chondrion is a granule-like substance. Mitochondria are small, granular or filamentous bodies which are present in the cytoplasm of all eukaryotic cells. Is also known as powerhouses of the cell. The first discovered by Colliker as a granular structure is present in the striated muscle. The other structural features of mitochondria are a double membrane bound organization is an energy converting organelle is present in all eukaryotic cells. The mitochondria are the major sites of aerobic respiration. The mitochondria is also known as powerhouse of the cell. The another important points are the mitochondria are a semi-autonomous organelle. Why is called for semi-autonomous organelles means they have their own genetic material. Next, the morphology of mitochondria. The morphology is the size, length, shape and number. The average size of mitochondria is 0.5 to 2 micrometer in diameter. The length is approximately 7 micrometer long. The shape of mitochondria is varies in shape. Is like club, racket, vesicular, ring or round shape. Next, what are the numbers? The numbers is present in vary from cell to cell of mitochondria. In examples, the amphibians it contain approximately the 3 lakhs of mitochondria. Next, the mitochondria is located the cells with high energy requirements is example for sperm cells and muscle. The chemical composition of mitochondria. The gross chemical composition of the mitochondria varies in different animal and plant cells. Mitochondria are found to contain 65 to 70% proteins, 25 to 30% lipids and 0.5% RNA, a small amount of DNA. The lipid content of mitochondria are composed of 90% phospholipids. The major composition of phospholipids are lecithin and cephalin. The 5% are less cholesterol and 5% free fatty acid and triglycerides. The inner membrane is rich. One type of phospholipid is called cardiolipin. The major structure of mitochondria are outer membrane, inner membrane, intermembrane space, cristae and matrix. The structure of mitochondria. The mitochondria is made up of a outer membrane and inner membrane. The space between the outer membrane and inner membrane is called as a intermembrane space, and the center, the finger-like projection structure is called as a cristae, and small DNA molecules is also present in the center of the mitochondria, and the ribosomes is also present in the mitochondria. The mitochondria covered a membrane is called as a outer membrane. It's made up of a phospholipid bilayer is looks like fairly smooth. It quite smooth and has many copies of a trans protein is called as a porine, which forms large aqueous channels through a lipid bilayer. The major role of porins to allow the free passage of various molecules into the membrane space of the mitochondria. This membrane resembles to sieve that is permeable to all molecules of 10,000 Daltons or less, including small proteins. The inner membrane of mitochondria are freely permeable only to oxygen, carbon dioxide and water. The inner membrane is convoluted, forming folds is called as cristae in the matrix. The impermeable to many solutes due to high content of phospholipid are called cardiolipin. The cristae generally increases the inner membrane surface area. The two faces of membrane is refers to as matrix side, another one is a cytosolic side. The matrix side is called as N side, the cytosolic side is called as P side. The structure shows the cross section of mitochondria showing the various layers of cristae. Mitochondria are double membrane envelope in which the inner membrane divides the mitochondrial space into two distinct chambers. The one is outer compartment, the another one is inner compartment. The outer compartment or perimitochondrial space or the intermembrane, the space between outer membrane and inner membrane. The space is continuous into the core of the crest or cristae. The inner compartment, the inner chamber or matrix space is approximately 80 to 100 angstrom, which is filled with a dense, homogeneous, gel-like proteinaceous material is called as mitochondrial matrix. The major enzymes include involved in the synthesis of nucleic acid and proteins, fatty acid oxidation and TCA cycle. Mitochondria have their own genetic material and the machinery to manufacture their own RNA and proteins. A human mitochondrial DNA sequence revealed 6,569 base pairs, it encoding 37 genes, 22 tRNA, 2 rRNA and 13 peptide genes. The inner membrane of mitochondria having a C phase and M phase. The outer cytosolic phase is called as C phase and inner matrix this phase is called as a M phase. The M phase attached to the inner mitochondrial membrane are repeated units of stacked particles are called as elementary particles. Inner membrane subunits are oxysomes. They are also identified as F1 particles. This means to produce the synthesis of ATP and also the ATP oxidation. The major enzymes are present in the mitochondria are the mitochondria is made up of a outer membrane, intermediate space, inner membrane and matrix. These all the parts of mitochondria is present in a certain enzymes. The table shows the major enzymes are present in the different part of the mitochondria. The outer membrane is contain the cytochrome B5, cytochrome B5 reductase, fatty acid CoA synthetase, fatty acid elongation system, phospholipase A, nucleoside diphosphokinase. The intermediate space contain the adenylate kinase, sulfite oxidase, nucleoside diphosphokinase. The inner membrane of mitochondria contain the cytochromes B, C1, C, A and A3 and NADH dehydrogenase, succinate dehydrogenase, ubiquinone, electron transferring flavoproteins, vector ATP synthase, beta hydroxy butyrate dehydrogenase, carnitine palmitoyl transferase, and the inner matrix of mitochondria is contained pyruvate dehydrogenase complex, citrate synthase, aconitase, isocitrate dehydrogenase, alpha oxoglutarate dehydrogenase, malate dehydrogenase, fatty acid oxidation system, ornithine transcarbamylase, carbamoyl phosphate synthetase 1. Next, the major functions of mitochondria. The major parts of mitochondria are play a key role in a energy transducer of the cell. It means the synthesis of ATP. Is also they play the important cycles are a Krebs cycle in matrix, electron transport chain, and the phosphorylation, and the storage and transport of ATP it produces as a result of cellular respirations are liberated through a transporter is called as adenine nucleotide translocase. The mitochondrial enzymes are also required for the synthesis of lipids are present in the mitochondria, and also the production of a heat to maintain a thermogenesis. The mitochondria are plays a major site of several metabolic reactions. The outer membrane it contains the major site of oxidation of epinephrine, degradation of tryptophan, and elongation of fatty acid. The inner membrane of mitochondria are a major site of oxidative phosphorylation, and the matrix of the mitochondria involve the major site of Krebs cycle, beta oxidation, detoxification of ammonia in urea cycle, and storage of calcium ions. The mitochondria also they play a key role in a apoptosis and synthesis of estrogen and testosterone. Is also act as a important role in a neurotransmitter metabolism, and role in cholesterol metabolism. It also play very important key role in a heme synthesis.